Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience-based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go. Let's make great science together.
Okay, so think about most cell therapies today. They basically recruit some kind of tool to a diseased cell, right? Like sending a delivery truck.
Delivering a payload.
Exactly. But what if you didn't just deliver? What if you could actually build a brand new functional protein, say a toxin or maybe a cytokine, right there on the cell surface? And crucially, what if that whole manufacturing process only started if the cell showed a very specific, like complex combination of markers?
That's a pretty counterintuitive idea, moving from delivery to, well, on-site fabrication.
It really is. And that shift is the breakthrough we're diving into today. We're looking at this synthetic biology platform called SMART that's splicing modulated actuation upon recognition of targets. It's from a recent Nature paper.
Right. It's essentially an autonomous decision-making device built out of proteins.
Yeah. It uses proximity, basically, to create these really quite sophisticated protein logic gates. So we're moving beyond just, recognizing one thing.
Like marker A.
Right. To doing actual Boolean logic, like only trigger an output if you see marker A, and D, marker B together.
Which addresses a huge problem in targeted therapy. Specificity.
Exactly.
Because, if you look at current methods, antibody-drug conjugates, CRIT cells, they usually go after just one surface antigen.
Yeah.
One target.
And that's often not enough.
Well, the paper really highlights this single receptor. It hardly ever defines just one specific cell type, especially in complex tissues or tumors where things are really messy and heterogeneous.
Right. You get a mix of cells.
You do. And relying on just one signal, that often leads to off-target effects, hitting healthy cells.
Which means toxicity.
Exactly. Systemic toxicity, which limits how much drug you can give, how effective it can be. It's a major bottleneck.
Okay, so the challenge isn't just finding the right neighborhood. It's making sure you're acting only on the exact house, confirmed by multiple signals. But how do you make sure a powerful protein, like a toxin, doesn't just wander off if the targeting isn't perfect?
That's the core innovation here. The missing piece was really a way to synthesize the effector protein in situ. Right there, on the spot.
Build it where you need it.
Build it only where you need it. From inactive parts. That gives you spatial control. And the tech enabling this is conditional protein splicing, or CPS.
Okay, CPS. How does that work?
So imagine the protein you want to build the output is split in half. The SMART system uses these engineered protein segments called caged split intimes.
Intames. Those are the self-splicing protein bits, right?
Precisely. They can cut themselves out and stitch the flanking parts, the exteins, together. Yeah. Here, they've engineered them so you have two halves, NTN and NTC.
Okay.
And critically, they're caged, meaning structurally blocked somehow, so they're inert on their own. Just inactive building blocks floating around.
That inactivity is key. So how do you activate them? How do you unlock the cage?
You force the two halves together, physically. Proximity is the trigger. When the two caged halves are brought really close, it forces this structural change, a sort of domain swapping event that forms the activin fold.
And then it does its splicing job.
Then it activates, cuts itself out, and ligates the flanking pieces, the actual output protein halves together into one functional molecule.
Right there on the cell surface. Okay, let's walk through their first proof of concepts, the AND gate. How did they set that up?
So figure one of the paper, basically.
Right. They designed two separate inactive polypeptide fragments. Each one has one half of that cage split in time.
In-time cage or NT case.
And importantly, each half is fused to a different targeting vector, like something that grabs antigen 1 and something else that grabs antigen 2.
Or DARPins in this first case, I think.
Yeah, DARPins. So these two fragments are just floating around harmlessly.
Until
they encounter a cell that has both antigen 1 and antigen 2 on its surface. Then the targeting vectors grab hold, pulling the two fragments together.
colocalizing them.
Right, colocalizing them. And that physical proximity, that's the trigger. It forces the intine halves to activate, splice, and ligate the output pieces together.
Creating a brand new functional protein, but only on that specific cell.
And the output they chose for this first test was really clever. Smart spycatcher.
Yeah, spycatcher is great for this. Once it's made, this spycatcher 003 protein, it acts like molecular superglue.
Superglue?
Basically. It spontaneously forms a covalent bond with its partner peptide, SpyTag, super stable, high affinity. It creates this perfect little dock on the cell surface.
Easy to detect then.
Very easy. And you can attach pretty much anything you want to the SpyTag later on.
And the results really showed that AND gate working. They targeted cells based on having both HER2 and EGFR.
Right, using K562 leukemia cells, some engineered to express different combinations.
And only the cells that were double positive, HER2 and EGFR positive, actually made the spycatcher dock and grabbed onto a fluorescent SpyTag probe they added.
You could see it light up, but only on those specific cells.
Yeah. And the crucial part, the wild-type cells, or the ones that only had HER2 or only had EGFR.
Nothing. No signal.
No signal, zero response, which really proves that rigorous AND gate selectivity.
Right.
The tool is only built if both inputs are there.
It's a very clean result. And once they had that fundamental AND gate working so well, they started showing how adaptable it is. Tunable, even.
Tunable? How so?
Well, they looked at the intine structure itself, the inter-GJ1 intine they actually got a crystal structure.
Oh, cool.
Yeah, and based on that, they made specific point mutations within the cage structure. Things like K114A, K116A.
And what did that do?
It dramatically boosted the system's sensitivity. They got up to like 150% more SpyTag recruitment, which tells you the response dynamics aren't fixed. You can actually engineer them.
Which is essential if you want to use this therapeutically, right? You need that control over the dose response.
Absolutely. Then they moved on to more complex logic.
Beyond just AND gates.
Yeah. Or logic was pretty straightforward. Just mix multiple targeting fragments together so you could get an output if either HER2 or EGFR was present.
Okay, that makes sense. But what about not logic? That seems harder. How do you tell it not to activate if a certain marker is there?
That was a really smart design. They introduced a decoy protein.
A decoy?
Yeah, so this decoy also had one of the intine halves fused to a targeting vector for the not antigen, let's say EGFR in this case. But crucially, the intine cage on this decoy was engineered to be a bit weaker, a bit more reactive.
Ah, so it's like faster to splice.
Exactly. So if a cell has the two ND antigens like HER2 and EpCAM, but also the not antigen EGFR, the decoy anti-EGFR preferentially grabs and splices with one of the main input fragments.
Blocking the desired reaction.
And essentially blocks the formation of the intended output protein. It's like shunting the system down a dead end if that third unwanted marker is present.
Wow. Okay, so that lets you do things like HER2 and EpCAM, but not EGFR. That's getting really specific.
Super specific. And they immediately showed the power of that precision by applying it to targeted cell killing, logic-gated cell depletion.
Right. So how did that work? Same spycatcher dock?
Same principle. The smart spycatcher gets made only on the cells matching to the complex logic. Then they add SpyTag, but this time the SpyTag is linked to biotin.
Biotin. So you can stick streptavidin to it?
Precisely. They added streptavidin linked to saporin, which is a potent toxin. The whole complex gets internalized, and the saporin kills the cell.
But only the cells that built the dock in the first place.
Only those cells. And the results were impressive. They got like 78% depletion of the target cells in one mixed population.
While leaving the others alone.
Yep. Single positive and wild-type cells were unharmed. And for one specific cancer line, A431, I think, they hit up to 92% depletion. It really shows the therapeutic potential of this level of selectivity.
That's incredible precision in a complex environment. And you mentioned earlier it's not just limited to standard antibody-type targeting.
No, and that's another really key aspect of the input modularity. They showed this works with all sorts of targeting molecules.
Like what?
Well, obviously the DARPins we mentioned, but also single-domain antibodies, SCFV fragments, smaller things too, even peptides. They used one called BKT140 that targets CXCR4.
Okay.
And even small molecule drugs. They use one called SCH58261, which targets a GPCR, a DOR-A2A.
So it's not just big protein antigens. You can target based on receptors that bind small molecules to.
Exactly. It really opens up the range of biological targets you could potentially go after. It's not just the usual suspects.
And the output side is just as flexible, right?
Yeah.
You mentioned the spycatcher dock is like a platform.
Yeah, they call it logic-gated proximity labeling. Because the dock is only built on the logic-positive cells, you can use it to deliver other tools very locally.
What kind of tools?
They showed delivering an enzyme, APISX2, which can be used for proximity biotinylation.
To map nearby proteins.
Right. Or they delivered an iridium photocatalyst. The point is, these labeling tools only become active on the cells that satisfy the input logic, like AG1, AND, AG2.
So you can map the immediate molecular neighborhood, the microenvironment, but only for that very specific subpopulation of cells defined by your logic gate.
It's incredibly precise proteomics, basically, restricted by the synthetic logic.
Okay, that's powerful for research. But let's talk about that IL-1 example. That seemed like a big leap.
It really was, because up to this point, with SpyCatcher, the output protein stays stuck, tethered to the cell surface, ligation and retain.
Right.
But what if you want the output to be released to act on nearby cells, like a secreted signal?
You mean like a paracrine effect? Sending a message to neighbors?
Exactly, like activating nearby immune cells, perhaps.
Yeah.
So they engineered a ligation and release version.
How'd they do that?
They basically just changed the architecture, the domain arrangement. Instead of having the output protein fragments flanking the intine, they put the targeting vectors flanking the intine halves, and the output protein, IL-1 in this case, was positioned differently.
So when the intine spliced itself out?
It released the fully formed IL-1 into the surroundings rather than leaving it attached via the targeting vectors.
And interleukin-1 beta. That's a pretty potent cytokine, right? And kind of complex structurally?
Oh yeah, it's a pro-inflammatory powerhouse and has this complex beta-barrel fold. It's not trivial to produce correctly, but the system did it.
And it only produced it when the right target cells were present.
Correct. For instance, on HER2 positive, EpCAM high cells. And crucially, they showed the released AL1 was biologically active.
How did they prove that?
They did co-culture experiments. They had the target cells producing the IL-1 via SMART, and nearby they had reporter cells, just plain HeLa cells. And they showed that the IL-1 released by the target cells caused NF-kappa B, a key signaling molecule, to move into the nucleus in the neighboring HeLa cells.
Which is the classic sign that the IL-1 receptor pathway has been activated.
Exactly, a clear paracrine signaling effect. It proves the SMART system can synthesize and release active, complex signaling molecules, but only where the logic dictates.
Okay, so stepping back, the big picture here. The core innovation seems to be creating the synthetic biology platform that's not just finding cells, it's acting like a protein actuator based on this conditional splicing.
Yeah, it fundamentally shifts how we think about engineering cell surfaces. We go from just, recruiting a static tool.
Right, sticking an antibody on.
Right, to actually having programmable dynamic fabrication of a new function triggered by multiple inputs.
And the implications, they seem huge for diagnostics.
You could design probes that only become visible, only light up, when a really specific complex pattern of disease markers is present. Imagine identifying very rare, very specific cell types with that kind of certainty.
Unparalleled specificity. And for therapy, this seems to directly tackle that systemic toxicity problem we talked about.
Absolutely. You can generate potent things, toxins, cytokines, but locally, right at the disease site, like inside a tumor. The Boolean logic confirms this is the spot, and then the weapon gets built.
So you confine the action, drastically reducing the collateral damage that plagues so many current treatments.
Exactly. Less systemic exposure, potentially higher efficacy right where it's needed.
And they mentioned the intine itself is quite tolerant.
Yeah, the NRDJ1 split intine they used seems to tolerate a wide variety of sequences being fused to it or being created by the ligation, which suggests that, in principle.
You could make almost any protein this way, provided you can split it.
That's the idea. Any protein that can be split and then re-ligated could potentially be an output.
Yeah.
And that opens the door to using different orthogonal intine pairs.
Meaning pairs that don't interfere with each other.
Right. So you could potentially build multiple layers of logic gates on the same cell surface. More complex circuits.
That's the real mind bender, isn't it? Programming the cell surface to not just see one thing, but to sense multiple signals, perform logical operations, and then autonomously build something complex as a result. It feels like a quantum leap in how we can control cells.
It really does. And if this tech lets us program a cell to read complex logic and build a specific protein right where it's needed, well, the next question has to be, how complex can we make these circuits? How many layers of A and D, R, not logic, can we realistically stack before we essentially turn the cell surface into a functioning biological computer?